With this image, we can start to talk about the signal transduction. And as an example, I will bring to you three major protein kinase systems that are present all over the species and all over the organism and should be for your interest in any time. We have to keep that the activation of the kinase, the protein kinase, come from the membrane. All the kinds of stuff that arrive in the cell is perceived by receptors, okay, that will trigger intracellular signaling cascades that will allow that cell to answer to that stimulus. So, this is a question that we may do. What is cell signaling? What's the meaning of cell signaling? Okay? The meaning of cell signaling is not so complicated. All the events, all the intracellular events that take the cell to answer to a stimulus that arrive. Okay? And it can be a biochemical molecule, it can be a hormone, it can be a peptide, it can be a physical stimulus, can be light, can be the temperature, okay? Everything that arrives on the cell and make the cell change the metabolism to answer to this kind of signal is called signal transduction, okay? One of the first types of signal transduction can be represented by that image, the cell-to-cell contact. So one cell contact to other cell, and they transmit each other some different signals that can be interpreted and that can generate an answer to that cell-to-cell contact. But not only cell-to-cell contact, the cells may talk with molecules, okay? So we can identify different structures, different molecules, proteins, peptides, lipids, cytokines that are involved in long-term communication of different cell types. So here we have two types very clearly. In the synapse, we know that there is the neurotransmitters that are released, and they are in abundant, okay? Very, very abundant here, and the receptors are in the side. So the synapse is a very small place with lots of signaling molecules, with lots of receptors. So the meeting of the receptor with its ligand is quite guaranteed, okay? But in the other side, you can see the production of an hormone by a cell or by an organ, by a glandule, and this will go through our bloodstream and will achieve cells far away from the place where it was produced. But that molecule produced in another organ can be interpreted in the other organ as a signaling molecule, for example, a hormone, as is depicted there. This kind of communication can be divided into the paracrine or the autocrine model of communication, okay? The paracrine mediators, they cannot go very far from the place where they are produced because they are labile. They are rapidly metabolized by the organism. So they are not good signal molecules for long distance, okay? They just play a role in the vicinity of the place where they are produced. And even these molecules, they can trigger signal in the same cell it was produced. It's somehow strange, but it occurs. More recently, there is another type of cell communication which is called the endocrine model, where the cell produces a signaling molecule inside it, and the receptor and all the cell machinery to interpret that molecule is inside that cell also. So it's not released. The signaling molecule is not released. It is a cytosolic product that is interpreted in the same cell. For the signal transduction, we have a very easy structure. We need a receptor, we need a signal molecule, we need proteins that are involved in the answer, and we will have products that are very famous, like the second messengers, that are molecules that in normal situation are quite absent in the majority of the cells, but when they are triggered, their concentration raises very fast. And if their concentration raises fast, the cells start to produce or to active different cell pathways to answer to that signaling molecule that arrives. The receptors, the first step, are the principal way to achieve protein kinases. The protein kinase can phosphorylate different amino acids, and the protein phosphorylation is one of the mechanisms, one of the most studied mechanisms to regulation of different enzymes and procedures in cell signaling. And there is a not correct correlation, because we used to think that when some protein is phosphorylated, it is activated. But it's not true. You can phosphorylate to turn it off, some kind of procedure. The kinases, they can phosphorylate at least 3 different amino acids: serine, threonine, and tyrosine, which is not represented here. So we have the serine, threonine kinases, and we have the tyrosine kinases. Okay? The tyrosine kinase we will see in more detail. The reaction is quite simply like this. You have the protein in the presence of ATP. The kinase transfer the gamma phosphoryl from the ATP to the protein, and that's it. The protein is now phosphorylated. All the protein kinases have a partner or a rival, that is the protein phosphatase. Okay? So for each phosphorylation, we have a phosphatase to dephosphorylate that substrate and return things to the homeostasis, to the normal situation of the cell. Okay? Just to you start to think about, there are lots of pollutants, there are lots of toxic protein molecules, substance that interfere with either protein kinases and phosphatases. One of the most toxic products of cyanobacteria, some cyanotoxins, are inhibitors of protein phosphatases. So if you inhibit the protein phosphatase, the substrate stays phosphorylated for a period longer than it was necessary. And this is a problem for the cell, okay? If you can identify these products which are phosphorylated for longer periods, you can infer, you can try to think what is the real problem in that situation, yes? You can try to bypass that problem and try to activate another signaling pathway. That's why it's somehow complicated. But if you put in mind that you can associate the environmental pollution, the ecology, the interspecies with cell signaling, we can try to improve the understand of this kind of thing, okay? That is the tyrosine. So we have groups of kinases that are serine threonine kinases, and we have tyrosine kinase. So, but it's somehow strange, because if a kinase is a serine threonine, it can phosphorylate a serine or a threonine in all the proteins? No, there is a very huge control on it, okay? The first control is when the cell decides to differentiate. When the cell differentiates, it starts to produce proteins and enzymes and substrates that are only found in that kind of cell, okay? So it is a specialized cell now that, upon a stimulus, has his own repertoire of cell signaling molecules. Another way of control the phosphorylation and the answer that cell will give is that the primary structure of the protein, the amino acid sequence, are quite different for the serine threonine kinase and for the tyrosine kinase. The kinases, is something like they read the protein, and they know exactly if that serine or that threonine is inside a sequence that they understand as a phosphorylable serine or a phosphorylable threonine, okay? If it's not in this kind of sequence, the kinase can be active, but it will not phosphorylate that protein. So there are very different types of regulation that I will not enter in the details because of the time, okay? The phosphorylation can directly alterate the activity of an enzyme. So if an enzyme is turned off, you phosphorylate it and it starts to function, okay? Or the contrary, if it is function and you phosphorylate it and it stops to function. So it's easy nowadays because we have different tools in the computer, in different sites that you can look to your primary structure of your protein and try to find phosphorylation sites on it and try to study what kind of pollutant, what kind of molecule could interfere with that. Another possibility, an enzyme, when you phosphorylate some protein, it's a harbor to adapt to other proteins, okay? So there are some domains in proteins, different proteins involved in cell signaling. They have a domain that binds to a tyrosine phosphorylated. So when a tyrosine in a protein is phosphorylated, it is now a harbor to different proteins that can attach to this other protein. And this start to build a puzzle, start to build something like a complex, a multi-protein complex that is involved in the cell signaling pathways, okay? These are the SA2 domains, okay?